So what I want to do is I want to do a review of the cell. I want to take a part of the cell and I'm going to expand it and blow it up. If I made that part of the cell membrane larger, it actually is comprised of a lipid bilayer. When we talk about the phospholipid bilayer of the cell, the construction is of a headpiece that is hydrophilic and tail pieces that are hydrophobic. To take a more detailed gander at this molecule, it's actually constructed from a glycerol molecule, a couple of fatty acid chains. Matter of fact, I think this one has a double bond and it kind of jukes sideways. The headpiece here is made out of choline and phosphate. The choline has a positive charge, phosphate negative, and this interacts well with water, which is a polar molecule. So the outside of the cell membrane and the inside of the cell membrane, inside and outside, both can interact well with water, but the internal portion of the membrane itself, these are comprised of hydrophobic fatty acid tails. Also in membranes, there will be protein channels. And protein channels allow certain ions into and out of the cell that are too big to normally move. Some chemicals are too large to be normally transmitted through the cell membrane. For instance, glucose is one of those. And in the case of glucose or other large molecules, there might need to be a protein channel. The protein channel will change conformation to receive the glucose, and then it'll actually change shape one more time to transmit the glucose through the cell membrane. Typically, one of the things that we see is that things move from a high concentration to a low concentration. That's called diffusion. So the simple process of moving things from high to low concentration is called diffusion. Normally, water moves wherever it wants to go. So if water's on the outside of the membrane and it's less concentrated inside, it just automatically moves to make the concentrated side less concentrated. One of the things that we say is wherever Mary goes, the lamb is sure to follow. Mary is sodium and the lamb is water. So anytime there's sodium in a given area, water moves to it. That's why people get high blood pressure, by the way. They eat a lot of sodium, water goes into the blood vessels to make the sodium less concentrated and the blood pressure rises. That's part of how that works. Not necessarily in a good way, but that's how your body works. It's called physiology. So if a membrane protein is used for movement, it is called facilitated diffusion. If we have a lot of a certain item on this side, and we want to even pump that one the opposite direction, in effect uphill, there is a way to do it. And that is called active transport, and it requires ATP to fit and unlock this channel so that the channel can move something against the gradient. Normally things in the cell move from high to low concentration. It's like moving from the top of a hill down to the bottom of the hill. Normally things at the bottom don't automatically and spontaneously move back to the top. Can you do that? Yes, you can do it, but the process is called active transport. So if there is energy involved, it is called active transport. And always remember that active transport requires ATP. I match those two together. A, active, A, ATP. Requires energy to do that. So the cell membrane is made out of a phospholipid bilayer, and the fact that it has fats in it is what makes your skin so soft. That's why in the wintertime, whenever your skin dries out, you put greasy, fatty stuff on your skin to nourish it. Why? Because that's what your skin's made out of. That's what nourishes it. It's the same thing. let's go back over to the cell and let's talk about the nucleus for a few minutes, because this is really the control center. And the nucleus is double membraned, as I'm drawing, and these membranes have holes in them called nuclear pores. They also have channels which lead out to things called endoplasmic reticulum. And so I'm going to label this, here is the nuclear membrane. By the way, we are talking about the cell. And when I mean the cell, I mean just a typical animal cell. We'll talk about the differences of an animal with a plant in just a few minutes. Notice that the nuclear membrane has pores. Why are these important? Because messenger RNA copies the DNA and then goes out onto ribosomes. Let's do a ribosome here, a few ribosomes, and it can actually translate proteins from a copy of the DNA. In the nucleus is the genetic material. We'll make some red and some blue here. And we will also make a little round nucleolus. The eolus means little nucleus, and this is where RNA is formed, and RNA is what manipulates the DNA. So inside, we say that chromatin is the uncondensed form of DNA. And then whenever it condenses down to these little X-shaped thingies to make chromosomes, then we call it chromosome. We don't call it chromatin anymore. If the messenger RNA makes a copy of the DNA, it can go and read it through a ribosome on the rough endoplasmic reticulum. This is a ribosome, and this is my little messenger RNA strand. Let's make the protein purple. We'll say that the protein is purple. And then what can happen in a really interesting way is this formed protein, from the process of translation, by the way, can bud out and then leave in a vesicle to go over to the Golgi apparatus, where final packaging occurs. The Golgi apparatus is very similar to the endoplasmic reticulum, but it is a final processing center for items made inside of the cell. So as you can see, once again, instead of leaving, this one is arriving. So the protein will arrive, let's put it right here, and the membrane of the little vesicle will actually fuse, let's call this a vesicle, will actually fuse with the membrane of the Golgi apparatus, and then this protein will move all through the Golgi apparatus and then be packaged on the opposite side. So it will come out over here eventually by an outpouching of the Golgi membrane like this. And so in the end, it will be a usable, modified package for either storage inside of the cell or for shipment somewhere else. Now, let's talk about that for just a moment, because this vesicle to me doesn't look any different from that one, but let me tell you there is a difference. This vesicle has not been processed yet, so this is like a Campbell's soup can in your hand. You could even close your eyes and know the size. You could think there's probably chicken soup in this, because there's not many cans this exact size. The truth is, though, once this Campbell's soup can gets processed through the Golgi apparatus, then it has a red and white label on it. All of a sudden, there's an ingredient list on here. So it has kind of reached a stage of usefulness. It can either be stored inside of the cell for use later, or it can fuse, pay attention now, it can fuse with the membrane, and this protein can be transmitted to the outside. That process, by the way, is called exocytosis. So even though I didn't show you the process of copying the DNA is called transcription. The process of making a protein is called translation. The protein is formed, it is modified, and it's completed, and then the protein can actually be transmitted outside of the cell to be used somewhere other than the cell. There's another thing that can happen, so pay attention to this, very interesting. What if there is a bacterium outside of the cell, and this is a white blood cell, for instance? Well, what is crazy is it could do a couple of different things. It could either go use cytoplasmic streaming to go and capture that, which is probably what happens, or it could just invaginate. This could collapse backward like this, and the bacterium could be basically sucked into the cell. It can be sucked into a vesicle, similar to the protein vesicle. And then what happens here, class, is a lysosome would attach to it. And the lysosome, with its digestive enzymes, make those digestive enzymes, would then cut this bacterium up so that the bacterial parts could even be used inside of the cell. So this process of capturing and eating something is called phagocytosis. Everybody say phagocytosis.
Phagocytosis.
And it's actually a form of endocytosis. Now there's one other modification that I want to make. What if there was a certain molecule shaped like this, and the cell wanted only that molecule? It could actually make an advertisement for that and place a receptor that's specific to that chemical so that it would capture that chemical, and then this whole thing, once again, would invaginate inward. Let's do like that. There we go. And this piece right here would collapse inward and carry this captured chemical inside. This would then pinch off and then form a vesicle that has captured only the desired chemical. This is called receptor-mediated endocytosis. Can spell. Endocytosis. Receptor-mediated, that means that's referring to this little piece that's picking up the custom chemical. Endo means inside, exo means outside. So remember that. Exo is when a cell is moving something out, and endo, inside. There's actually another form of endocytosis, and it's called pinocytosis or pinocytosis. That's a case where the cell can drink. Instead of eating something, in effect, it can drink something. So pinocytosis is another flavor of endocytosis. There is endoplasmic reticulum that doesn't have ribosomes on it, and that is called the smooth endoplasmic reticulum. By the way, that was a ribosome, the site of protein synthesis. The smooth endoplasmic reticulum is called smooth because it doesn't have any ribosomes, and by the way, this is where fats are made. So they would be processed the same way. They would be sent through the Golgi apparatus, and they could be packaged and sent somewhere. So fats can also be produced by the cell. One of the big organelles inside of the cell that is double membraned is actually the mitochondrion. I'm going to draw this a little bit larger than actually a lot larger than it should be. I'll kind of give you a scale in just a minute. If you looked at the mitochondrion, normally it would be about, compared to the nucleus, it would be about this big. It would be like a little caterpillar compared to the nucleus. We're talking about little, but I'm kind of drawing it a little bit larger so that you can see what's happening. And so glucose is captured into the cell, and I actually drew right where I need some room. And then the glucose is then brought in, is split into two, into pyruvate. And then these pyruvates are brought into the cell in the form of acetyl coenzyme A, and then they're taken to a cycle called the Krebs cycle. So the first step, which is kind of jumbled here, is called glycolysis. And I'll have another video that breaks this down and explains it in greater detail. But the glucoses are broken into pyruvates. Pyruvates have some CO2 stripped off of them, and then acetyl coenzyme A is a 2-carbon fragment that then mixes with a 4-carbon fragment to form a 6-carbon fragment. A couple of CO2s are stripped off of this, and there are a lot of energy producing compounds that will carry the energy from these chemical reactions to what's called the electron transport chain. Those energy carrying molecules are B vitamins, riboflavin and niacin. There are also ATPs formed along the way, so that in the end, in the electron transport chain, when oxygen is used as the final electron acceptor to make water, in the electron transport chain, oxygen is used as the final electron acceptor to make water, one of the waste products of your body. You have to go to the bathroom. That's some of the water made in cellular respiration. In the process, in this final process, when we look at the final count, your body will get 36 to 38 ATPs per molecule of glucose. That is called aerobic respiration. And as I said just a minute ago, I will break that down in greater detail in other videos. Some people believe that the cell obtained mitochondria as primitive bacteria that were power cells out there, and that the original protocells brought these mitochondria in, and then they were replicated along with the cell and eventually became housed with the cell. In an evolutionary scheme, that is called the endosymbiotic theory. Now, there is another person called Michael Behe that has an idea called intelligent design. And what he says is that you can't piece by piece a cell together. It's like a mousetrap. It either has all of the parts that it needs to function or it doesn't. And so there's a little bit of and I think maybe a lot. Some books written back and forth about why this person is right and that person is wrong. But a couple of different theories on how primitive cells became more complex. Of course, one group would say that this happened by capturing other organisms and then using them in the cell, and then one group would say that it was designed. A couple of different viewpoints to look at. One of the important parts of a cell is called the centrosome. If we look at the centrosome, it's made of two little things that look like Twizzlers to me. Each one of these is called a centriole, each one. And these are what will move from one side of the cell to another to kind of choreograph the movement of chromosomes in mitosis. Pretty interesting how that works. There are a lot more things that are accomplished by the centrosome and the centriole, the internal framework of the cell, microtubules. So much is accomplished by these, and I probably don't have even enough knowledge to tell you all the things, but I do know that they can transmit, these tubular systems can transmit hormones, their pathways. I even learned in one video, this is crazy, that the mitochondria can move on tubular networks inside of the cell like caterpillars. They can go where apparently the energy is needed. Unbelievable. So in one of the videos I want to show you later, the cell is called a city. And that's one of the things that I'm seeing is that this is so incredibly amazing. It is organized like a city. It is hard for me, in my mind, to conceptualize all of this as a disorganized accident or an accident, a series of accidents. It does appear quite amazing. There are about 3 differences between an animal cell and a plant cell that I like to talk about. And so let's do that for just a minute. Number 1, a plant cell has an extra cell wall. That's one thing. And then secondarily, there is a presence of chloroplasts. There are chloroplasts inside of a plant cells. These are stacks of thylakoid discs. So chloroplasts. There are also large central vacuoles in plants. Central vacuoles I originally thought were just where sugars that were formed by the cell were stored, but I have since understood that pigments are contained in these central vacuoles that give a plant its color. Also poisons, like poison ivy, the poisons that make you itchy, are also located in these central vacuoles. So when we talk about plants, these are the 3 big differences that I want you to be aware of. There are some other organelles. There are peroxisomes. There are certain, like a lysosome, there are certain vesicles that help break down other cellular metabolites. There are also some cells in your body. If you're a male, you have cells that have a flagellum. That would happen to be in a sperm cell, spermatocyte. There are also ciliated cells. Some cells in your body have cilia, especially the ones in your lungs that are built to help you cough up junk out of your lungs so that you don't just collect all kind of trash down there. So it's a matter of convenience, but could be life-saving as well. After thinking about what we've already looked at here, there are a few modifications that I want to make. Number 1, ribosomes can be found freely in the cytoplasm, and they can actually be found as polyribosomes. They can actually be long chains of ribosomes. And what is so cool about it is several ribosomes can actually write or read the same messenger RNA code and make multiple proteins at once. That's kind of cool. Multitasking is one of the abilities of your cell. So you can have a polyribosome. It's possible to have free ribosomes. That's a possibility. Another thing to think about is that everything inside of the cell is called the cytoplasm, and the liquid itself is called the cytosol. So the cytoplasm is just everything, everywhere inside of the cell. I had that messed up in my mind for a long time. There is actually a word for the liquid. I actually have a video from the 70s or the 80s that calls this cytosol the cell sap. One other last thing. Earlier I said that it's possible that the chloroplast might be double membrane. It's not only possible, it's actually true. Chloroplasts are double membrane, just like the mitochondria, and the thylakoid disc inside of the chloroplast also are bound by membranes as well. So a few extra little items that will increase your level of understanding of the cell.